Good evening, class. I hope you are all doing well. Hello.
Hi.
Hi. Hi.
Hi, we are all fine.
Okay. So let's start with the nervous system as we discussed last week. So the nervous system is the system that controls almost all activities of the human body. And therefore, you look at how it functions, and then subsequently, we look at its influence in the various nervous systems. So upon completing the first model of the nervous system, you should be able to locate and identify the cellular components of the nervous tissues that make up the entire system. Then you should know the structural function of the nerve cell or the neuron. Then you should also be able to explain the formation of the resting membrane potential and also the action potential. And we look at how transmission of impulse occurs along the nerve cell. Then finally, after completing this model, you should be able to explain the factors that is involved in the transmission of action potential from one neuron to the other, that's across the synapse. So the nervous system is one of the most complex systems within the human body. And it's complex because it's made of some specialized cells called neurons. And we mentioned neurons during introductory physiology, where we said it functions as relaying signals, conveying or perceiving, and also transmitting signals to and fro in the body. And we have the brain as the main organ of the nervous system that helps with coordination of actions and also processes life activities. So the nervous system is responsible for everything from sensation, movement, and all organ functions to learning and keeping memory as well. So we can categorize the functions of the nervous system into three based on the structural components of the nervous system. So we have three main types of nerves or the nerve cell. You can have the sensory nerves. And when I say nerve is a tissue made of neurons or the nerve cell. So whenever I mention nerve, I'm talking about the tissue, not the cell. When I want to talk of the cell, I'll include the cell as such. So we have the sensory nerve, which is tissues that are made of nerve cells that transmit signals from parts of the body through the spinal cord to the brain, which is the main region for coordination of activities. So the sensory nerves is responsible for transmitting signals from parts of the body through the spinal cord to the brain. So at the end of sensory nerves, we have receptors, sensory receptors, that senses change. So the change can be external or internal stimuli. So when we're talking of homeostasis, we mentioned that we have receptors or sensors that will sense a change that happens whether from the external or internal of the body. These receptors are sensory nerve endings that would sense the change. And then through the sensory nerves, a signal is sent to the spinal cord and the brain for coordination. So the first function of the nervous system is to sense change and also transmit signals through the sensory division to the brain for coordination. Then you have the second function of the nervous system as integration. Integration mainly happening within the central nervous system, which is the brain, and also the spinal cord, but mainly the brain, where we have most activities interpreted or analyzed. So we have the sensory information coming from the sensory division, reaching the brain, which is the integrative center, integrating or processing the signal, and eventually bringing out a response. Now, the response will come through the motor nerves or the motor division. So that's the third function of the nervous system, where we have transmission of responses or command from the integrative center to parts of the body for an activity to occur. So the motor nerves or the motor division is made of nerves that is connected to from the spinal cord or the brain to parts of the body that is transmitting command or responses from the central nervous system to parts of the body for an activity to occur. We can still use homeostasis to explain these functions, where we have sensory nerves transmitting a signal to the brain. The brain interprets the signal and bring out a command to effectors. So now the brain is the integrative center, serving as the integrative function. And then we also have the command coming through the motor nerves, which is the third function, transmitting responses to effectors for an activity to occur. So we look at the cellular components of the nerve tissue. When we talk of the cells that make up the nervous system, we talk of the neurons. The neurons. The neurons are the functional unit of the nervous system. So the neurons are the functional unit of the nervous system. When we say functional unit, that's where the activity of the entire nervous system begins from. So that's the cell that is responsible for the functional aspect of the nervous system. So we have each neuron responding to and also transmitting and communicating to another neuron when there's a change. So as we move on, you will look at how neurons communicate with each other, and that's how you tend to transmit signals from one neuron to the other. So we say we have three types of neurons based on their function. So I've already explained this based on their functions. We have the sensory neurons transmitting signals from parts of the body to the CNS or the central nervous system. We look at the interneurons, which remain in the CNS, that is going to help with processing and storage of information. Then we have the motor neurons that will transmit feedback or transmit responses or command from the central nervous system back to the body. So looking at the structure of the neuron, a specialized cell, specialized because of the structure and its unique function, which is transmission of signals in the body. We have the branch-like structures, which are the dendrites that receive signals from nearby neurons or even from parts of the body. Then we have the cell body or the soma, which is where the nucleus and other organelles are found, and that is where the cell is kept alive and is kept functioning. And we have the axon hillock, that is the neck extending from the cell body, continuing with the axon. And it's the axon hillock that generates, that's the point where a threshold is reached to generate an action potential along the axon. We'll talk more about it. Then we have the axon, which is the elongated part of the neuron. That's where transmission of action potential happens. Then we have the final part, which is the axon terminal, which is the end of the neuron terminal. And this is where there will be another neuron connected to transmit the signal to. Aside these parts, you can also have some of the neurons having myelin sheaths. These myelin sheaths are specialized cells that help increase the speed of the transmission of the signal. So the myelin sheaths are insulators, meaning they can't conduct the electric signal that is passing through the axon, and therefore it speeds up by the signal jumping over each of these myelin sheaths, and that hastens the transmission of signal along the axon. Then we have the second type of cell that is found within the nerve tissue or the nervous system, which is the neuroglia or the glia cells. The neuroglia or the glia cells. These are cells that support the neurons, and it provides a common example I just mentioned was the myelin sheath. So the myelin sheath is formed by cells within the central nervous system. The myelin sheath that forms the neuroglia cells that form this myelin sheath is the oligodendrocyte. Then within the peripheral nervous system, we have another myelin sheath that also another neuroglia cells that also form this myelin sheath, and that is the Schwann cell. So the Schwann cell, the oligodendrocytes are all examples of neuroglia cells that support the function of the main neuron or the nerve cell. So we have these cells that are providing support. In the central nervous system, we have the astrocyte, we have the ependymal cells, oligodendrocyte, microglia cells, all providing their unique support to the neurons so that they can help transmit the signal effectively. Then we also have the peripheral nervous system, which is also having two types of the neuroglia cells, that is the satellite cells and then the Schwann cells. These two are also performing their own unique functions to support the neurons within the peripheral nervous system. We look at membrane potentials. Membrane potentials. Any questions so far? Any questions so far? Yes, Joshua.
Please, sister, with the types of the nerves, the neuroglia cells, that one I don't get it. Please, does it mean that it is part of the neuron cells?
Okay, all right. So The neuroglia cell is another type of cell within the nervous system. You know, then it's a system. A system is made of organs. Organs are made of tissues. Tissues are made of cells. So we look at the foundation of the nervous system, the cells that are found within the nervous system, and that's what we are looking at. The main one is the neuron. The neuroglia cells or glia cells are supporting cells. They are also cells on their own. So the ones you see like this, forming the myelin sheath, you can see that there we have the round circular representation inside this round cell. That is the nucleus. Okay. So they are cells on their own found within the nervous system. Just like how we have this entire neuron. This is an example of a supporting cell, but here their support is to speed up the transmission of signal along the axon, and therefore they form myelin sheaths. We have other neuroglia cells, like what I mentioned. The astrocytes, for example, found in the spinal cord and the brain, help maintain blood-brain barrier, provide structural support, regulate ion, nutrients, and dissolved gas concentration. So each of them are separate cells that are supporting the neurons. Okay, and they are providing their individual support to the neurons wherever they are. So when you talk of the cells that are found within the nervous system, is the neurons, the main one, and then the neuroglia cells. Joshua, are you clear?
Please, yes, thank you.
Okay. All right, membrane potential. Now, when I was talking of the transport across the cell membrane, I made mention of sodium ions, potassium ions. I also made mention of the sodium potassium ATPs as a primary active transporter. Now, across the cell membrane, we have a resting membrane potential. A resting membrane potential is the electrical difference or the difference in charge across the cell membrane. And the charges are as a result of the different ions we have in and out of the cell. Our emphasis will be based on two main ions, the sodium and potassium ions. Sodium ions are higher in concentration outside the cell than inside. And then we also have higher concentration of potassium inside the cell compared to the outside. And I'm sure you remember when we were talking of how sodium potassium ATPase work. We mentioned that we have the sodium potassium pump. It pumps 3 molecules of sodium out the cell and 2 molecules of potassium inside the cell. Now, I also made mention during our introductory physiology that we don't have equilibrium of these ions and substances across the cell membrane, in that you don't have the same concentration of sodium in and out, or you don't have the same concentration of potassium in and out. There's always differences. And since these are ions, they carry charges. So the differences in charges across the cell membrane will give you a membrane potential. Now, we have the first one, which is the resting membrane potential. And this is the membrane potential that is found across the cell membrane when the cell is at rest. When the cell is at rest, it means it's not functioning or it's not performing its function. When you take the neuron, since we are talking of the nervous system, we talk of the neuron. The neuron functions as to transmit signals. So when the neuron is not transmitting any signal, we have a resting membrane potential around the cell membrane. Now, whenever the neuron starts to transmit a signal or even receives a signal that needs to transmit it and then pass it to another neuron, then we have the resting membrane potential changing to either graded or action potential. I'll take that again. We have differences in charges because of the differences in ions across the cell membrane. When you say across, meaning the outside and inside. Sodium is in higher concentration outside than the inside. Potassium is of higher concentration inside than the outside. We have other ions. You can talk of calcium ions, have a lot of calcium outside than inside. You can talk of chloride, a lot of chloride inside and outside. So we have different ions in different concentrations, and that creates a differences in charge across the cell membrane. That gives a membrane potential. And at the resting state, when the cell is not functioning, when the neuron is not transmitting any signal, there's a resting membrane potential of the neuron. So we say at resting membrane potential, voltage-gated sodium channels are in their resting state, which are closed. Voltage-gated potassium channels are also closed. So we mentioned channels as the means through which ions are transported. We have types of channels. We have gated channels. So the gated, let me see if I have a slide. Yes. So these are the different kinds of ionic channels that we have. So we have first one, the leaky channels. Leaky channels are always opened. They are leaky, meaning they are always open, transporting ions. So we have sodium leaky channels. So we have sodium. I need your maximum attention here, please. We have sodium leaky channels. We have potassium leaky channels. All the ions you can think of across the cell membrane, we have their specific channels, leaky channels. And these leaky channels are always open. So when you talk of sodium leaky channels, they are always open, leaking sodium from their higher concentration to their lower concentration. Leaking sodium from their higher concentration to the lower concentration, meaning sodium will move from the outside into the inside of the cell. Potassium has a leaky channel, potassium leaky channels, leaking potassium from their higher concentration to their lower concentration. That's from the inside to the outside of the cell. Then we have ligand-gated channels. And these are channels that would only open when a particular ligand or chemical binds to it. When a particular ligand or chemical binds to it. An example is what happens across the synapse. We'll talk more about this. So we will have neurotransmitters, or for a common one, acetylcholine. That is a chemical that will bind to a particular receptor on the cell membrane, and that would enable the carrier protein to open for the neurotransmitter to enter the cell. So that's an example of a ligand-gated channel. Then we have mechanically gated channel, and this opens with force, any mechanical pressure, force, stretch, anything that can touch, even a touch can open these gated channels. Then finally, and the one that our focus is on, is the voltage gated channels. These channels open when there's a change in voltage, when there's a change in the membrane potential. Whenever there's a trigger where the part of the neuron moves from its resting state, then these channels open. We'll talk more about it. So let's look at the voltage-gated channels. Now, all these channels I made mention, they are found within the cell membrane. They all act accordingly. So when voltage-gated channel, or ideally when the cell is at rest, when the neuron is at rest, the voltage-gated channels are closed. You know, they are gated. They are gates on them, and they are closed. They are not like open, the leaky ones. So if these voltage-gated channels are closed and there is a change, a little change in, let's say, the dendrites of the neuron, a little trigger that would change the membrane potential, let's say from minus 70 to minus 69 or minus away from its resting state, then this voltage gated channel would open. Thomas, let me call you back. I'm teaching. Let me call you back. So I'll take that again. So when you have these channels are closed, whenever there's a change in the membrane potential such that it's not normal, or when there's neuron starts to transmit or starts to receive a change or a stimuli, this voltage gated channel is going to open. And it will open at the area or location where there's a change in membrane potential. Now, you say neurons communicate with one another through electrochemical signals, and these signals are in two. We have the graded potential, then the action potential. Now, graded potential occurs over a short distance, and normally it happens across the dendrites and then the cell body. It happens along the dendrites and then the cell body. Then we have action potential. The action potential is the actual transmission of signal along the axon of the neuron. So there can be instances where a neuron receives a signal from the dendrites, passes the signal down to the cell body or the soma. And there is a possibility that it doesn't reach the axon. So if it doesn't reach the axon, this graded potential that was generated dies out. But if it's able to hit a threshold at the axon halo, then an action potential is generated across the axon. I'll explain more. So we say the formation of graded potentials and action potentials are dependent on the existence of the resting membrane potential and the presence of many ion channels that provide the path for ions to flow across the cell membrane. So before any neuron can receive a signal, it should be within its resting state. It should be within its resting state. So even after a neuron has transmitted a signal, it needs to return to its resting state before it can receive another signal. Now let's take it one after the other. Resting membrane potential. Now the resting membrane potential, in summary, states or explains that the inside of the cell is negatively, it's more negatively charged than the outside. So the inside of the neuron, so we are talking of the nervous system. So then the inside of the neuron is more negative than the outside. The inside of the neuron is more negative than the outside. That doesn't mean the outside is not negative, but the inside is more negative than the outside. And that doesn't also mean that the outside is positive. We are just comparing the charges in and out, and therefore we say the inside is more negative than the outside. So at the resting state, the neuron, the inside of the neuron is 70 times negative than the outside. The inside of the neuron is 70 times negative than the outside, and therefore we have the optimum resting membrane potential of the neuron to be minus 70 millivolts. Okay. So now, if the inside of the cell is more negative than the outside, what accounts for this change? We have, first of all, the maintenance of the resting membrane potential through the sodium-potassium ATPase. We mentioned that the sodium-potassium ATPase transports 3 sodium out, 2 potassium in. So if 3 plus out, 2 plus in, meaning there is -1 inside, and you have a lot of these sodium-potassium ATPase transporting sodium and potassium accordingly. And therefore, within the cell, you have negative environment compared to the outside. Then you also have movement of cations through leaky channels. Now, movement of cations through leaky channels, we have still focusing on the sodium and then the potassium ions. Now, potassium leaky channels have the ability to leak a lot of potassium compared to sodium. And if potassium is if potassium is being transported at a faster rate, meaning you tend to have a lot of positives leaving the cell compared to positives coming in. I hope that is clear. So if we have sodium and potassium leaky channels, all transporting sodium and potassium accordingly, you tend to have a lot of potassium leaving because potassium leaky channels transport potassium at a faster rate compared to sodium. And that also makes the inside negative, more negative. Then we also have trapped anions within the cytosol or within the cell. These anions, they are proteins, molecules that carry a negative charge that are found within the cell. So these are the factors that would contribute to the negative environment, the more negative environment within the cell. So let's take note that the plasma membrane or the cell membrane is said to be polarized when it is in its resting state and depolarized when it is active. I'll explain this. So a resting cell is said to be polarized. When we say polar, it carries a charge that is -70 millivolts, and I've explained what account for this charge difference. Now, I'll explain the depolarization as I move on. We have two types of responses that can be elicited if a neuron is stimulated. Now we are moving away from the resting state. Whenever there's a change or whenever the neuron detects a signal, a change, the state of the neuron moves from its resting state to either graded or action potential. Now, the type of response will depend on the intensity of stimulation. Very, very important. So if the intensity of stimulus is not high, the stimulus can be generated within the dendrites, and then the signal can be generated within the dendrites and then the cell body and remains there, eventually dies out. If the intensity is high, then it can cross to become an action potential along the axon. Now, let's take graded potential. Any stimulus that opens a gated channel produces a graded potential. So a gated channel, so voltage gated channel can generate a graded potential, or even a ligand gated channel can generate a graded potential. So we say a stimuli occurring at the dendritic nerve endings trigger the opening of mechanically gated or ligand gated ion channels, which enable a local flow of ions across the plasma membrane, producing a graded potential. Let me use the transmission across the synapse to explain this so that it becomes clear. Now, let's look at this picture. This is one neuron and this is the second neuron. So we have transmission. We have transmission happening from this neuron to this neuron. So the second neuron is the postsynaptic neuron. The first is the presynaptic. Now, we zoom in into the area where the neuron 1 is going to transmit a signal to neuron 2. So we want to look at how a graded potential is created at neuron 2. We want to look at how the resting membrane potential of neuron 2 is going to be changed. Or we still want to look at how this neuron 2 will receive the signal and transmit it. At rest, the inside here is minus 70. The membrane potential along this cell membrane is minus 70 millivolts. So the space between neuron 1 and neuron 2 is called the synapse. So within the synapse, there will be some chemicals released. They are called neurotransmitters. I'll explain more when we get to this topic. But these chemicals are the ball-like structures you see. They are neurotransmitters, and they have their specific receptors on the neuron 2. So they will bind to the neuron 2, and that is a change that the cell membrane will recognize, and that would trigger the opening of some voltage-gated channels for ions to rush into the cell, changing the resting state into a graded potential. Okay, so we go back. Now, it's the same thing. I've put it in writings here. So when there is a stimuli along the dendrite, it triggers the opening of ligand-gated channels. So the ligand, which is the neurotransmitter, binds to the receptor, a specific receptor, and that's going to trigger some voltage-gated channels to open for some ions to enter. Now, whatever that will move the inside of the cell away from its resting state is either going to negate it more or is going to move it to a more positive value. All that I'm trying to say is the resting state is minus 70. So it's either we move it to minus 71, minus 72, or we move it to minus 69, minus 68. Now, when we are moving it to a more negative value, we are inhibiting, okay, we are inhibiting the transmission of signal. But if we are moving the value to a more positive value, that's minus 69, 68, then we are exciting the cell. That means we are trying to ensure that we transmit the signal, we propel the signal ahead. Okay, so graded potentials are changes in the membrane potential that cannot spread far from the site of stimulation, elicited by the stimulus of subthreshold strength. All that I'm trying to say here is that we have the transmission, the signal received at the dendrites, So this is the dendrite to receive it and then spread this signal across the dendrites and then the cell body. That is graded potential. But whenever it crosses the axon hillock, we all know the axon hillock, to the axon, then it turns into an action potential. So with graded potential, we have depolarization and hyperpolarization that basically explains deviation from the resting membrane potential. So depolarization, as I explained, is where we move the membrane potential to a less negative or more positive value. And hyperpolarization is where we move the resting state to a more negative value. Now, with depolarization, the inside of the cell becomes less negative or more positive than the resting state due to the influx or the transport of more positive ions. So if you have -70 and you move more positive ion, let's say you move +10, that means it becomes -60. Then you move +10, it becomes -50. We are moving to a more positive value. Then with hyperpolarization, we say we make the inside more negative by moving negative ions into the cell. So here we don't move positive, we move negative ions. So it can be chloride ions where we move negative ions into the cell, making the inside more negative. And it becomes difficult for the cell to be excited. So what does graded potential do? It moves the resting membrane potential closer to a threshold for transmission across the axon. So depolarization that I'm trying to say is making the cell or the inside of the cell less negative to a positive after hitting the threshold. Then when you move it, the membrane potential to a more negative environment or negative value, we say the cell is hyperpolarized. So if a graded potential or combination of graded potential is strong enough to depolarize the plasma membrane from the resting potential to a threshold, which is about -55 millivolts, an action potential is generated. All that I'm trying to say is, let's still use this diagram instead to explain. Now, kindly follow. If you are sleepy, wake up. I need your maximum attention here. This diagram basically explains everything. Now, when the dendrite, which is the part of the neuron that receives a signal, when it receives a signal, you have the movement of positive ions into the cell. Then it will change the membrane potential from its resting state to a graded potential. So let's say around this dendrite, we have about 10 sodium plus entering. That means -70 plus 10, that will be -60. So by the time it gets to the cell body or the axon hillock, which is also known as the trigger zone, then the membrane potential should reach -55. If it gets here and it's -56, the signal cannot cross to the axon. It will remain as graded potential at the cell body dendrite and eventually die out. And this is dependent on the intensity of stimulation. Then, if the membrane potential gets to the axon hillock, which is also the threshold, the trigger zone, and hits a threshold of -55, then it propels along the axon as an action potential. It's either -55 is hit or not. If you reach -55.5 millivolts and you are not able to, yes, -55.5, meaning is more than the -55, you still can't be moved along the axon. The threshold is strictly minus 55. So it's either you reach the minus 55 or you die out within the soma or the dendrite. So you say the dendrite, that's the reception. Dendrite, you see the neurotransmitters and then and triggers voltage gated sodium channels. So there is influx of more sodium ions into the cell. Then that is the actual propagation of the signal along the dendrites and then the soma. So we have, since this is an entire cell, the cell membrane is full of channels, okay? And I mentioned the sodium and potassium channels. So wherever the signal gets to, it triggers voltage gated sodium channels to open for sodium to rush into the cell, changing the membrane potential as it moves on. And that is the actual transmission that we are explaining. So whenever we say how does transmission happens along the dendrites and then the cell body, we say there is a change at the receiving site as a ligand binds to its receptor. And that's going to trigger voltage-gated sodium channels to move from the outside to the inside of the cell. Then that will change the membrane potential around that environment. Then that change is going to trigger the subsequent voltage-gated sodium channels to open. Then sodium rushing, the membrane potential changes. Then that will also trigger subsequent voltage-gated sodium channels to open. Sodium rushes in. Then so it continues that way till it gets to the axon hillock. Now, when it gets to the axon hillock, the nature of the axon hillock is such that we have a lot of voltage-gated sodium channels within the cell membrane of the axon hillock. And therefore, when the signal gets here, mostly it should be able to hit the threshold where you tend to have a lot of the opening of these channels for a lot of positives to come in so that you can hit the minus 55. But if the intensity of the stimulus is low, such that it doesn't hit the threshold, then you would have the signal dying out here without moving to the axon. So I've put them in writings here. The graded potential that is capable of reaching the threshold minus 55 depolarizes or excites the axon. Hey. Hello, can you hear me?
Yes, please.
Yes, ma'am.
Yes, madam.
All right, okay. So before the graded potential can be propelled along the axon, we have two main ways. We have the temporal summation, we have the spatial summation. Where is that? Let's see. The temporal summation is where we combine two or more graded potential. Due to maybe their intensity not high, you can combine two or more graded potential from one neuron to ensure that a threshold is reached. Then you can also talk of the spatial summation where we combine two or more graded potential from many neurons to reach a threshold in a postsynaptic neuron. Then we look at action potential. Action potential. Now, when this graded potential is able to cross the threshold and reach the axon, we term it as an action potential. And whenever an action potential is generated, meaning the signal is able to cross to another neuron for information transmission. So action potentials are triggered by graded potentials, as I mentioned, and exist as either muscle action potential conducted through muscle fibers or nerve action potential. So we will talk of muscle action potential. You talk of muscle action potential in the muscular system. So you do only nerve action potential as we are doing the nervous system. So action potential is a self-propagating electrical potential difference produced across the plasma membrane of a neuron or muscle cell when they are stimulated. So this is basically what happens along the neuron when we have a threshold reached. So in the neuron, we say it arises at the trigger zone or the axon hillock, as I use the diagram to explain, and is propagated along the axon. Then we say propagation does permit communication over a long distance, and it's all or none, because it's either you reach a threshold or you don't reach, and therefore when you reach, an action potential is generated. When you don't reach, the graded potential dies out. Then it happens over a short duration ranging from 0.5 to 2 milliseconds. We have phases of action potential. Look at the latent period. The latent period is basically the time taken for the stimulus to travel from the point of stimulation to the recording electrode, basically from the dendrites, the cell body, to the axon hillock. Then when a threshold is reached, we have the opening of voltage-gated sodium channels along the axon, causing depolarization along the axon. Then we have the third phase, which is the repolarization. Repolarization is we are repolarizing, we are bringing back the membrane potential to its negative state. We are bringing back the membrane potential to its negative state so that it can receive another signal. And here, that means we have to move a lot of positives out so that we can get that negative environment within the cell again. And when we are moving a lot of positives out, we would move sodium out, because it's sodium that can move from a higher concentration outside the cell. So here, voltage-gated potassium channels are open. It's potassium I'm referring to. Potassium channels are open for potassium to move out of the cell. And as I said, the rate at which potassium moves out is faster than sodium. And therefore, there are instances where potassium, a lot of potassium will move out of the cell, making the inside of the cell more negative than the resting state, and that is hyperpolarization. So hyperpolarization is when we open voltage-gated potassium channels, then potassium will rush out of the cell, creating that negative environment inside the cell again. And as I said, because the potassium channels transport potassium at a faster rate compared to sodium, a lot of potassium will leave the cell, creating a more negative environment than the resting membrane potential. And when that happens, we have hyperpolarization. So hyperpolarization occurs, and therefore the closure of the voltage-gated potassium channels, the efflux of potassium through non-gated, that's the leaky channels, and also the main thing, the action of the sodium-potassium pump would work together to bring back the resting membrane potential, which is minus 70 millivolts. So the action potential is represented in the graph. So you have here, the down part is the time in milliseconds. Then the y-axis is the voltage. So the resting state of the nerve cell is minus 70. So at the resting state, then we have a stimulus. So this can happen at the dendrite. So when it happens at the dendrite, we have transmission happening at the dendrites and then the cell body. If the intensity of the stimuli is not that strong, then it can die out there. But if the stimulus intensity is high, such that it's able to reach the threshold of minus 55, then an action potential is generated along the axon, where we have the influx of sodium into the axon or the nerve cell. Then we have repolarization, where we need to move back there. So we have the peak of action potential to be plus 40 millivolts. Some books will also give you plus 30 millivolts. Then we have repolarization, where we need to move back the membrane potential to its resting state so that this neuron can receive another signal. And this will happen when we move a lot of positives out, creating that negative environment. And that is repolarization. Mainly it's potassium ions that move through voltage-gated potassium channels out of the cell. When that is happening, there are instances where the inside of the cell becomes more negative than the resting state. For example, minus 90. And that is hyperpolarization. So the work of the sodium potassium pump, closure of the gated potassium channels work to move back this membrane potential to minus 70, which is the resting state. Same diagram. So I think I'll use this to summarize both of them. Yes, a lot of potassium when you are talking of repolarization. Repolarization. All right, so in summary, we look at a signal received at the dendrites, and a signal is received when a ligand-gated channel opens for a ligand to bind to it. Then the complex form is going to trigger voltage-gated sodium channels to open for sodium to rush into the cell. So positives will get in, moving the resting state to a more negative or less negative, a more positive. Okay. So now, as the signal moves, we open more sodium, voltage-gated sodium channels. Sodium gets in, the membrane potential moves to a more positive value. So as this happens, it happens across the dendrites and then the cell body. When it gets to the axon hillock, which is the trigger zone, we have a threshold of We have a threshold of minus 55 reached, and that propels the signal along the axon as action potential. Now, action potential wave propagation happens along the axon, where you have the subsequent opening of voltage gated sodium channels. Then sodium gets in, same the membrane potential changes, then it will trigger subsequent voltage gated sodium channels open, and then sodium gets in till it gets to the axon terminal. Now, when this is happening, we have the repolarization also happening, where voltage gated potassium channels open for potassium to leave the cell so that the inside will return back to its negative environment. Now, at the axon terminal, the signal will cross a synapse to another neuron, and we'll talk more about that as we move on. So what happens around the dendrites and the soma is called the graded potential. When what happens along the axon is the action potential. When the neuron is at rest, it has a resting membrane potential. Communication between neurons. Any questions up to this point? Any questions? Any questions? All right, let's move on. Yes, Emmanuel. No, it's not Emmanuel. Bosiakon, Dennis.
Okay, sister. My question is a bit sorry for the word. The issue is when you were saying something about the resting membrane potential. I said what contributes to it. You made mention of the movement of calcium through the leaky channels. Please,
No,
that
cations. Cations.
Oh, okay. I thought it was.
Yeah, cations.
Oh, okay.
Movement of cations
Then
through leaky
thank
channels.
you.
All
Thank
right.
you for the rectification.
All right. Okay.
Oh, okay.
Yes, any other questions? We move on. Communication between neurons. Now, we have transmitted a signal from the dendrites to the cell body along the axon terminal. Now we are at the axon terminal. How does the signal cross from this axon terminal to another neuron? So how do neurons communicate with each other? So we say, sorry, I'm going to move the two bar down. So this is the means by which signals or impulses are transmitted from one neuron to the other. And communication happens by the release of hormones or neurotransmitters. Neuro is neurons, transmitters transmit. So these are chemicals that help transmit the signal across the synapse. Calisius, I'll come to you. Let me get to a point. So we have signal molecules that initiate this action by binding to their receptors and generating a response. Communication may be from a nerve cell to another nerve cell. That's what we are going to talk about. Then a nerve cell to a muscle cell, we talk about it in the muscle system. Then we can also have the nerve cell and then the gland. So the site of transmission of signal information is the synapse. The synapse is the space between one neuron and the other. The neuron that is transmitting the signal is the presynaptic cell. The neuron that is receiving the signal is the postsynaptic cell. So synapse may be electrical, found at gap junctions, or chemical, found at the synaptic cleft. So the presynaptic neuron, the postsynaptic neuron, between them you have the synapse. You can have some types of synapse. You can have the synapse between one neuron and the other. You can have the synapse between the neuron and the skeletal muscle cell, which is the neuromuscular junction. Neuromuscular, the neuro and the muscle. Neuromuscular junction, the neuroglandular, the neuro and then the gland cell. You can also have different synapse based on the parts that is connecting. You can have the axosomatic synapse, the axon terminal of the neuron 1, then the soma of neuron 2. You can have axodendritic, the axon terminal of neuron 1, the dendrites of neuron 2, axonal, the axon terminal of neuron 1, the axon of neuron 2. So when you look at the chemical synapse, well, that's what we are going to look at, because within the body we have a lot of these compared to the electrical synapse. So the chemical synapse releases chemicals into the synapse, and it's these chemicals that will help transmit the signal within the postsynaptic neuron. Then we have the electrical synapse. The electrical synapse directly transmits the electrical signal through gap junction channels. So these are channels that are found that holds the two neurons together, still with the presence of the synapse, but you have these proteins that are channels that hold these neurons together, and they help transmit the signal directly through to the postsynaptic neuron. Most of the synapse is the chemical synapse, and therefore we look at how transmission would occur. Now, I need your maximum attention here. So with the transmission of signal, across the dendrites, the cell body has graded potential, hits a threshold at the axon hillock, then transmission continues as an action potential along the axon. When the signal gets to the axon terminal, when the action potential gets to the axon terminal, first of all, it triggers voltage-gated calcium channels to open. Calcium, since we have a lot of calcium outside than the inside, calcium will move from their higher concentration outside to the inside of the cell. The presence of calcium inside the cell is going to trigger synaptic vesicles within the axon terminal containing neurotransmitters to move to the surface of the cell membrane. With the presence of a protein called docking protein, these vesicles grow an ability to fuse with the cell membrane, releasing their contents, which are the neurotransmitters, into the synapse by exocytosis. These neurotransmitters bind to their specific receptors on the postsynaptic cell, and then the complex form is going to trigger voltage-gated sodium channels to open. Sodium will rush into the cell, changing the resting state of the cell here. Then that is going to trigger subsequent voltage-gated sodium channels to open for sodium to enter. And that creates a graded potential on the postsynaptic cell. I'll take it for the last time. When an action potential gets to the axon terminal, it triggers voltage-gated calcium channels to open. Calcium enters the cell. The presence of calcium within the cell triggers vesicles containing neurotransmitters to move to the surface of the cell membrane with the help of a protein called the docking protein. The vesicle fused with the cell membrane, releasing their content, which is the neurotransmitters, into the synapse by exocytosis. Neurotransmitters will bind to their specific receptors on the synaptic cell, the postsynaptic cell. Then that complex form is going to trigger voltage-gated sodium channels to open. Sodium will move into the cell, moving the resting state to a more positive or less negative. So let's say minus 70 to minus 60, 69, 68, and that is graded potential. So subsequent voltage-gated sodium channels to open for that to continue. So you have graded potential transmission along the dendrites and the soma. When it gets to the threshold, minus 55 is reached, then voltage-gated sodium channels along the axon they are also open for the influx of sodium, moving the membrane potential to around minus 40 along the axon. Then when it gets to the axon terminal, this repeats. Any questions? Callistus, your hand was up. I've put them in writings here. When you get the slides, you read.
Yes, Madam, just a little clarification. I'm confused about something. And the generation of the action potential that you have just described, is it applicable to the three types of neurons stated, or each of the neurons has its own peculiar mechanism of generating the action potential? Is it a generalized mechanism?
It's a generalized mechanism. And in fact, it's not only the nerve cell. Okay. For when you talk of the sensory, this is happening. When you talk of the neuron, where we are transmitting a response from the CNS, this is happening. The interneurons too, we have this happening for processing. And it's not only the nerve cell. You can talk of muscle cells. When you get to the muscle system, you talk more about it. You also have the influx of sodium that is generating an action potential. Within the cardiac muscle cell, we have action potential. Within the skeletal muscle cell and smooth muscle cell. So we have these muscles as excitable that have the ability to receive a signal and also for the activity to occur. So this is a general the general transmission happening within all the neurons.
Okay, thank you.
You're welcome. Yes, Salisu Shaban. Salisu, your hand was up, or? Okay. So let's look at neurotransmitters. We have different kinds of them, what they do. They are chemicals that are released at the end of a neuron upon the arrival of an impulse to help transmit the impulse to another nerve cell, a muscle, or any other structure. We have neuroexciters, neuroinhibitors. Depending on what is released, we have a particular activity, whether excitation or inhibition of an action potential. You have them there. When you get the slides, you look at them. So we will end the class here for today. I think I've said a lot, and I'm sure you are processing them. Get the slides together with reading materials and digest them well. God willing, next week, I have a funeral. I'm hoping to come around early. So if I come around, I'll prompt the class rep. Shouldn't you, let's take it that you have the class. So if there's anything, I'll prompt the class reps ahead. Okay. So kindly read ahead. Module 2, look at the central nervous system composition, the brain, function, regions, parts of the brain, what they do. Then we look at the peripheral nervous system, and so on, the reflexes. Then we finish with the nervous system. We continue with the muscular system. Thank you very much. Any other submissions? Any other contributions? Any other questions? All right. If there are no questions, we end the class for today. Thank you very much. Oh, there's a hand up. Finoria.
Madam, please, good evening.
Good evening.
Thank you for the lecture. Please, when are we getting the slides, please?
It depends on the class rep. They should just hit me. I'll send it to them.
Okay, thank you.
You're welcome.